Greetings, science aficionados, and thanks for tuning in to our latest episode of Friance Sci. We're your hosts, Carlos and Fiona, and today we will explore how certain plants have developed particular photosynthetic pathways in order to thrive in hot and dry environments. How do they accomplish this? Also, as humans prepare for a rapidly changing climate, what is the relevance and role of these photosynthetic superheroes?
Let's begin with an overview of C3, C4, and CAM plants and their photosynthetic pathways. In C3 plants, initial carbon fixation occurs via Calvin cycle enzyme rubisco. Such plants are called C3 plants because the first organic product is a 3-carbon compound. Around 85% of all plant species on the planet are C3 plants, such as rice, wheat, soybeans, and most of the trees. What happens when C3 plants are exposed to hot and dry conditions? Initially, the stomata are open to allow CO2 to enter the plant, but under hot and dry conditions, evaporation intensifies, meaning that too much water is lost. C3 plants close their stomata to avoid hydraulic failure, but then CO2 can no longer enter the plant. Under low concentrations of CO2 in the chloroplast trauma, rubisco is capable of binding O2 in place of CO2 in a wasteful process called photorespiration. Thus named because it occurs in the light, photo, and consumes O2 while producing CO2, respiration. However, unlike normal cellular respiration, photorespiration uses ATP rather than generating it. And unlike photosynthesis, photorespiration produces no sugar. In many types of plants, including a significant number of crop plants, photorespiration drains away as much as 50% of the carbon fixed by Calvin cycle.
C4 and CAM are alternate modes of carbon fixation specialized to decrease photorespiration and retain water. How do they do this? They use carbon concentrating mechanisms, or CCMs, to ensure that the troublesome rubisco enzyme is exposed to high concentrations of CO2 and low concentrations of O2. These plants have two carbon fixation reactions. The first involves an enzyme that fixes CO2 into a 4-carbon molecule. This molecule can then be transported and converted back to CO2 and then fixed a second time as part of the standard Calvin cycle. The C4 and CAM cycles differ in the separation of these two carbon fixation reactions. For CAM plants, they're separated temporally. Carbon is first fixed at night when the stomata can open and not lose too much water as carbon dioxide enters the cell. The stored 4-carbon molecule is then processed in the Calvin cycle during the daytime when light is available for photosynthesis. For C4 plants, the two stages are separated physically in a pattern of inner and outer cells known as Krantz anatomy. Carbon is first fixed in the outer layer of cells, the mesophyll cells, that are exposed to the air. The plant then sends the stored 4-carbon molecule to the inner cells, called bundle sheath cells, for processing in the Calvin cycle. Here, there are low levels of oxygen, and this is key. You can think of this as the plant's way of filtering the air and selecting just the carbon dioxide to send to these isolated inner cells.
By reducing photorespiration, the carbon concentrating mechanism allows C4 plants to maximize the carbon assimilation and produce more sugar per unit of air entering the stomata. C4 plants thus thrive in hot environments and still obtain the necessary carbon while at times keeping the stomata closed to save up water. CAM plants take water saving to the next level. By opening their stomata only at night, far less water is lost through the open pores, leading to a high level of carbon assimilated per unit of lost water. This is known as water use efficiency and can be 5 to 10 times higher in CAM plants than in C3 plants. But nature's genius doesn't stop there. Introducing the C3 CAM hybrids. Plants can exhibit an entire continuum of phenotypes ranging from weak CAM to full CAM, and there are plants that undergo facultative CAM. they carry out C3 photosynthesis under normal water conditions and only shift to the CAM pathway of nocturnal CO2 intake and storage under low water conditions. These specific adaptations allow a plant to maximize CO2 intake via the C3 photosynthetic pathway when water is plentiful and still avoid carbon starvation or hydraulic failure when water is scarce.
When we talk about climate change, the catchphrase is greenhouse gases. And rising CO2 levels alone will benefit C3 plants to a much higher degree than C4 plants, which already concentrate CO2 at the site of the rubisco enzyme. In essence, the performance gap is lessened because higher atmospheric CO2 concentration decreases the inefficient C3 photorespiration. In reality, though, climate change is not just CO2 in the atmosphere. Rising temperatures and decreasing precipitation are imminent, which has severe implications for C3 plants that lack adaptations for these conditions. So as C3 plants experience water stress and less carbon intake, they will be outcompeted by CAM and C4 photosynthetic powerhouses. As climate change progresses, warm biomes will expand into higher latitudes, and the C4 plant range is predicted to expand accordingly. To address the threats that climate change poses to C3 crops, scientists are proposing to bioengineer C3 crop plants to undergo CAM or C4 photosynthesis. Some of the most promising prospects involve integrating facultative CAM pathways into C3 crops. This option would allow plants to maintain high levels of CO2 intake when water is plentiful by sticking to the C3 pathway. Or, if we engineer the C4 pathway into C3 crop plants, we could potentially equip them to withstand increasing soil pollutants, because a recent study indicates that C4 plants have a higher tolerance for nanoparticle pollutants due to their specialized krantz anatomy. But engineering the krantz structure into C3 leaves poses an additional challenge. Thanks for joining, and see you next time.